All right, I'll assume we're recording, and I can't see online, so if there are questions, are you following on Teams online by any chance?
I'm not.
You're not. Oh, okay.
You guys see it?
Maybe I can keep this. Okay, I'll keep this laptop here, but I can maybe see questions. All right, I'll try to follow questions on this. All right, afternoon. I'm Paul Taylor. I'm the director of the Scientific and Statistical Computing Core. We're also known as the AFNI group for the neuroimaging software that we develop. And I'm going to talk a little bit about fMRI processing and some considerations with it. So can I just ask, who's actually used fMRI data here? Most? Okay, great. Is anyone going to be using fMRI data as part of their project? Okay, great. For everyone else, sorry, this is a waste of your time, but. Okay. For the recording's sake, there was laughter. All right. So what I want to do here is actually talk a little bit about, we'll get to looking at processing and some considerations of it, but I want to kind of start from the very beginning with just reminding ourselves what we're trying to do. So we want to study neurons. We're going to use magnetic fields looking at oxygen in blood flow to do that. And then there's some positives of this and then some challenges. And processing really straddles both, trying to accentuate the positive parts, mitigate or get around some of the negative parts. And so this is a cartoon picture of what we want to think about studying. So we're interested in brain activity. This is neurons firing. So you can just imagine cartoonishly, there's no activity, and then all of a sudden something happens and a neuron fires and it goes back to being off again, this very brief activity. This is the reality of what we want to study. We're not going to look directly at that thing. So you can do single photon recordings or things like that, or single neuron recordings. What we're going to do is we're going to look at the blood oxygenation response to that neuronal firing, or really a large group of neurons in kind of a couple millimeters or so spatial scale. And so the BOLD response, it's an acronym, blood oxygenation level dependent signal. And so if you see down here, the gray shows the neuron up and down instantly, basically, but the BOLD response is actually going to have a slight couple-second delay, then go up and down. And this is a very simplified version of what's happening. Different parts of the brain is probably, the details are different, but the main thing is this is a delayed, long, slow, extended response of something different than what we actually want to study. And we just want to keep that in mind as we're going through. We're looking at a very secondary measure to that thing that we want to learn more about, which is the brain firing. And this BOLD response is not actually something, we don't have a little hydraulic anything kind of measuring the blood pressure directly. We're looking at something secondary to the BOLD response. We're looking at its effects on the magnetic fields that are that we've been placed in the scanner. So we have a couple layers of secondary that I've mentioned here so far. Hemodynamics instead of neuron response, and using the magnetic field fluctuations based on that BOLD response. That's what we're going to try to study neuron firing with. And so just as, again, kind of a cartoon picture of a very simplified idea of what we think is happening here, that in the top row in blue, I put like a stimulus or input time series. So basically imagine that first one was the neuron firing I showed before, and then imagine that the neuron is active for longer and longer periods of time. That's what's being shown here. The fact that they're spread out in time doesn't matter. I just needed to visually separate them. The main thing is that they're the kind of same amplitude, so the same strength of neuron firing, they're just getting longer. You know, so it's like if you were saying you know, something like that, a longer response. What we expect in the BOLD response, so the blood oxygenation, is that the kind of one for one, if you look vertically there, they're matched. The BOLD response also gets longer in time, but it's also getting taller in amplitude up to a point somewhere around the mouse, somewhere around like here, it doesn't keep getting larger and larger infinitely, it hits a plateau. And the mathematics of how we connect this and the expected response from that kind of instantaneous one is called convolution. And it's just an idealized mathematical representation of that. But it's just, this is kind of what we would expect. Now, if the neuron firing itself increases, that would increase the amplitude. But here we're just talking about the kind of duration. And if you notice that, if you kind of look at how they're synchronized in time or not so much, in each case, whenever we start a stimulus and end it, the bold response is delayed from the start and it keeps on going afterward. You'll probably by about this 10 or 15 second lag or some fraction of that. And so this is kind of what we expect. So a little bit delayed, the amplitude can go up with the duration and kind of comes down later. Are there any questions? I'm also very happy to take questions during this. Please don't be shy. Yep.
So we want to tie basically neuronal firing to metabolic demand in some way. But the amount of oxygen that the body, your brain needs to replenish itself, there's a known overshoot with the bold response. So I'm just kind of curious where the field is on that.
So in terms of the actual HRF shape, does it tip that? Yeah. So sure. When I picked the bold response shape, I left it kind of very simple, just going up and then back down. But it's true, it may go up, overshoot down, and then come back up to zero. I think a lot of responses do that for very instantaneous things. When you have longer duration events, that undershoot gets a little bit washed out. We often don't worry about it too much. But yes, certainly for instantaneous responses, that would be a more accurate representation in general. But all these things vary a lot around the different parts of the gray matter. Yeah. Okay, so since you've learned so much about fMRI, so you're ready for a quiz here. Okay, so just picture in your mind, what do you think that the bold response would look like? And the idea is here, the axis I should mention down below is time. And so the idea of this is, if you put someone in the scanner and you do something to have their brain fire for these 4 different events of about 20 seconds each, what would we expect the bold response to look like from it? Okay, so just from what we looked at before, picture it. And did you picture something kind of like this? Again, all of my stimuli have the same height and the same width, so I expect the same shape to be appearing below. It's smoother, delayed, lasts a little bit over the edge for all these kind of features there. Okay, and so the idea is now we can play this game where if I give you a stimulus design, you should be able to kind of draw the bold response picture. Is everyone happy with that? Okay, good, because here's another quiz. So if I gave you that top and I said this was the stimulus pattern that we gave someone in the scanner, what would the idealized bold response be? Do you think it's A, B, C, or D? A vote for B. Any other? Okay. Well, yeah, I agree. So when we look through these, we can say, well, there's 5 events up above, so we expect 5 events here. They're all the same duration, so I expect them all to be the same amplitude. I can look for the start of each one, and all these features would lead me to B out of all of this. Okay, so I can give you one, and you can find it from all sorts of different possible bold responses. And actually, this game is particularly useful because this is really what we're going to be doing with task-based fMRI. So what I'm going to do is when we put someone in a scanner and we give them a task to do, we know the stimulus of what's happening and when. We control when does a stimulus start, let's say shining a light or showing them words, how long does it last for, and we keep track of that. And then what we're interested in is to say what parts of the brain are involved with this task. We're going to search through all the different regions of the brain and look for bold responses that look like what we would expect. So it's literally the same game. And we call that the idealized bold response. And the stimulus pattern is the thing that we give, whatever shining lights, reading, button pushing. We estimate our idealized bold response, and then we go look for it. And that's what we're going to think are the most active regions of the brain are most involved. Now, another wrinkle to this is that the real time series aren't just 0, nice smooth shape, and back again with nothing else happening. Hopefully, while you're in the scanner, your brain is involved in many kinds of activities. You're breathing, your heart is beating. All these things are making large, significant changes to your blood oxygenation constantly. You're at a non-zero temperature in terms of Kelvin, hopefully. These are all things that lead to this kind of noisy baseline here, okay? And what this kind of representation means is I'm showing you one axial slice of a brain. So like if I took a knife and just kind of went through my forehead backward and then opened my brain up, we're looking at that slice. And the yellow box is making a grid around my basic units that I'm measuring in, what are called voxels, and that's a 5 by 5 grid of voxels. And so what I'm doing is I'm saying in that 5 by 5 little unit of space in the slice, let me look at the time series that I've recorded in the MRI scanner across time. Okay, so this is a time series for one voxel, another time series, and if these gold lines show synchrony across all of them just for visual reference. Is that clear how this is looking? Now, when we look at these, just what do you see when you look at these time series? Obviously, nothing is 0, and there's what we'd probably call noisy, a lot of unrecognizable patterns up and down. Many of them are about the same height over time. There's a little bit of baseline tilting. Anything else you notice? Okay, now what if I tell you that, for the record, the answer was no. And what if I tell you that this was our idealized bold response for this stimulus design? So we put someone in the scanner, we gave them, in this case, this is an audio and visual combined task. Doesn't really matter, but there are these stimuli, these blocks of about, well, anyways, it doesn't really matter the duration, but this is the stimulus response. Do you see anything in the time series that might be related to any of this? Okay, yeah, not all of these time series are the same kind of noisy. When you see this kind of gives you a sense of what should you be looking for. If you sift through this, there are some of things down here that, sure, they don't look one for one like the same smooth thing, but you see this kind of on-off waviness or what we call boxcar shape. And indeed, these time series down here are ones that we would think just visually are maybe associated with that, right? It's very difficult for this to happen randomly. Any questions on that? Okay. And indeed, this is the more realistic version of the game that I showed you before. Real data don't just have nothing happening. You give a stimulus and now you get these shapes and you said that one, that one, that one. There's lots of other stuff happening here. So as I mentioned, there's more than just what we call neuronally based bold firing. There's breathing, heart rate. Subjects move in the scanner, and this creates a phenomenally difficult problem because the part that you're measuring now goes out of, to a different voxel. So now suddenly you're gathering data from somewhere else in the brain. This has magnetic field feedbacks and things like that. All of this is what we have to try to deal with in processing. So basically, we want to do this game while taking into account these kind of difficult features that are in our way. Okay, so where have we gotten to here? So neuron firing creates bold response, yay, we put it in magnetic fields, awesome. And now we have to provide the stimuli of something to measure. So this is one very large chunk of fMRI studies called task-based fMRI, where you provide these stimuli and then you go look for the responses that you would expect in some form. There is a lot of subtlety to it, but it's basically this kind of pattern matching. Okay, so some practical aspects of fMRI. We've talked about it a little bit. There's good things, not so good things, and of course, the ugliest. So let's start with the good ones. Really positive things. It's non-invasive, no cutting involved. It's non-ionizing, so you can get scanned day after day after day if you have the patience for it. Some people, I'm sure in this room, I don't know how many times Vinay has been scanned, but probably a fair amount, and he's perfectly normal. So, yeah. So you can do many different, we want to study brain function. There are lots of different kinds of tasks that are really important that you can do while someone is stuck even in a metal tube. You can do reading, language, movies, neurofeedback. People can push buttons and react to what they're seeing, all sorts of things, even just resting still. There's whole brain field of view coverage, so we can see everywhere in your brain. Other modalities like EEG or MEG are great, but you're basically getting stuff only near the skull. You can't really get into the more interior parts of the brain. And we want to study the functional parts of the brain, which are mostly the gray matter, and those have typically in humans about a 2 or 3 millimeter thickness. And the voxel size that we have for fMRI is roughly around, let's say, 2 or 3 millimeters, and there's a lot of flexibility in that, but basically that means we can stick our measurements of unit, spatially these voxels, into gray matter and get meaningful stuff out. It's not like a giant thing where gray matter is just passing through. On the temporal side, because we're studying function, so we can scan people over time, and we can get whole brain snapshots about every 2 seconds or so. And again, there's plus or minus on a lot of this, but we have what we call reasonable temporal sampling. So the data are very noisy, but you can still see patterns related to our task. So what do I mean by noisy? I mean that there's lots of bumps and wiggles all the time. And even here, if we kind of recognize from before maybe that similar on-off response we might expect, there's lots of other stuff here, but we see our main shape of interest coming through, even though there's lots of other waves involved. So that's this kind of balance of things. MEG and EEG have much higher temporal sampling of order milliseconds, so you get a lot more information per second or per scan. But again, these are all trade-offs. 1 to 2 seconds for a lot of tasks, that's reasonable. It's not 5 minutes, you get a picture, things like that. Good, okay, we've made it through all the good stuff. So here we go, buckle up. This is what time series often look like in a real practical scan. This measurement modality is extremely sensitive to people moving. People moving a millimeter is a very bad thing. Try not to move a millimeter where you're sitting right now. You failed, right? Okay, everyone, it's very hard to do. And especially while you're doing tasks, sometimes you're pushing buttons, parts of you are moving and things like that, you get uncomfortable. And what does motion do? It does a lot of different things. So geometrically, your brain is here and you're measuring everything, and let's say you have a voxel right here. If I move, what is this voxel now recording? Because the field of view stays here, right? The box where the measurement's taking place and I moved within it, now I'm starting to record from something totally different. Maybe air, maybe a different part of my brain, something like that problem. And just in terms of kind of time series features, it's particularly bad because it puts in features that are very unpredictable. So all this is, I've highlighted one column of time across 5 neighboring voxels. So 5 adjacent parts of my brain I'm measuring. Sometimes there's an up-down jump, a bigger one, nothing, a very big one, or a very big one. That's very unpredictable. And in other parts of the brain, it's just up or just down. In different times, it may go a different way. This is what I'd call a baseline change. So it's not even just up and down, it's up and it's staying on a new plateau. This is extraordinarily difficult to do real rigorous time series analysis with it. It's even worse than this because your head is partially magnetic. So you have a magnetic thing moving in a magnetic field that creates bad things. So there's a lot of different types of badness that go into this. And if you focus on where this arrow is here, there's a further kind of distortion where at the moment when you're moving, with fMRI, we are not just acquiring data, poof, instantaneously I have a whole volume. we actually spend that 2 seconds of getting the whole volume going through slice by slice. So as you move, measurements are taking place constantly, and you move, and now your whole rest of your measurements are thrown off as well in this kind of what we call interleaved pattern. So basically, it's very complicated things happen. You can't just rotate the brain back when you were moving during that time, because it wasn't being measured as a whole solid thing. It was being measured slice by slice. Okay, so yeah, here, this is a useful zoomed in. So when you're moving during the acquisition of a particular volume, you often get this particular bright, dark pattern, which is a hallmark of problems in being said. We're also using magnetic fields. I'm a physicist by training. I like magnets, but they have big challenges. The human brain did not evolve for MRI scanner use. We have very important parts of our brain that we want to study just kind of behind your eyes and eyebrows and moving back. Those happen to be just above your sinuses. Having a tissue air boundary and then using changing magnetic fields and gradients to try to measure things in the brain is very, very bad. It's very difficult. So this person's brain actually has, in real life, brain material here. When we acquire, there's kind of no data there because of magnetic field distortions. And there's lots of aspects of this. This is a particular one. Yeah, it's just due to the magnetic fields. And you know, if you don't have signal there, you can't recreate it magically. So you have to work with your scanner techs, treat them very, very nicely, and have them help refine sequences and maybe focus on different parts of the brain. This is another distortion. This frontal part of the brain was not kicked in any way. The person's brain is just a normal shape. This is a different kind of magnetic field distortion, dealing with the way we acquire data, quantum mechanics, I guess. And so the brain appears squashed in here. Again, you can't kind of unsquash the brain very easily. The information that should be here has been kind of mixed in down here, and you can't separate it out so easily. So different parts of the brain have very particular distortions and problems, kind of your run-of-the-mill sequences. And if you want to study these, you have to maybe do particular tricks to do it, to work around the quirks of magnetic fields. So now, that was just the bad. Let's get into the ugly. Oh, I mean the interesting, because that's what the processing people like our group kind of like to spend time doing. Well, anyways, so the interesting part about this is how do we use processing to try to get around some of these problems or try to ameliorate them. So we can't pretend like they were never there, but we can do things to try to help reduce their impact, or at least know when we should give up on a particular data set. So this is a particular processing step in fMRI, like why do we do what we do, is to kind of deal with all these things I just spoke about in the good and the bad. We want to accentuate, use the good parts, and try to reduce, minimize the bad parts. So that's why this is the interesting part. If you notice here, this is a video playing on the left. It's the brain kind of before correction. So this is just as the field of view sees the person. You can tell there's a nodding motion in there. And if you look at the time series, kind of from a few different voxels, a little 3x3 grid, you see that when that nodding motion happens, there's a big spike. A very common form of fMRI processing is called motion correction. What we do is we just take the picture of the brain, and we take one, let's say you've acquired 5 minutes of data, and you have 200 time points or something like that. You choose one as a special reference, and you geometrically align every other time point to that one. So if the person moved and rotated their head, this alignment step should put it back to where the reference one was. Just by doing that, you can see what happens to the motion, the spikes that are particularly due to motion. Some of them go essentially away in some time series, and in other cases, they're actually reduced. I know that this one looks taller than its counterpart here, but you have to kind of take it as a relative height. So the height relative to this amplitude here, it looks bigger here because the floor beneath it is bigger. So actually this has gotten better, believe it or not. So again, it doesn't solve all our problems, but it does help in some cases. It does also mean that, going back to this original problem where I have a voxel here that I'm measuring, now I moved and I'm measuring something else, I should be reconnecting my time series that should be here again in an ideal world. So for all these reasons, we perform this step of motion correction on the EPI datasets. We call it correction, we're lying to ourselves, it's not correcting it. It doesn't make it look as if there was no motion, but we like that term because it sounds perfect. If you look at the left, that's what an fMRI data set looks like as a single volume. And this is what a real anatomical brain of the person looks like. This is someone walking down the street you could probably recognize if you saw all the slices. That thing on the left, you're not going to do it because it's distorted, it's fuzzy, it's low resolution. And so when we want to try to do group analysis, what we want to try to do is identify the same part of the brain across all my subjects and then do something with that. So I want to identify the motor cortex in my brain, your brain, your brain, your brain, and do statistics on it. It's much more challenging to find the motor cortex in the one on the left or the putamen or whatever you care about. It's much easier on the right here. So what we do is we've acquired two different kinds of data sets from the same subject. The one with the functional data that we care about, doing the task, emotional responses, houses versus faces, whatever. We acquired this one here that I've now underlaid. Let me go back, because this one has good anatomical fiduciality. So they're kind of partner, and we're going to use the strength of this one being anatomical features to align that fuzzy distorted one. We're going to use alignment with some stretchability to the actual anatomical fiducial one to try to put things into where they should be on the subject's own brain. And then later I can try to align this that has more detail, the grayscale underlay one, to a standard space template. If I try to just align the one on the left that's fuzzy, low resolution, and a little distorted, I would not be as successful as getting the motor cortex directly to my template brain motor cortex as if rather than if I had gone through this one stop here in a good anatomical brain. Because this is the person's own brain, the features should be very closely matched, but this should help undo some distortions to kind of stretch and squash things appropriately. Does that make sense? It's literally a lot of hand waving up here, but this is the idea of the alignment process. Notice even in here, by this alignment process, if you look at this part of the brain here, basically the hot colors here are where I have pretty good signal, the cold is where it's almost noise. If you notice, you can just see part of the grayscale underbrain poking through here. There's no real signal here to use. I have numbers coming through, I have time series there, but they're going to be basically all noise. And that is, again, due to this geometric distortion of having sinuses. You can see the sinuses here. It's having these air pockets right around the brain. That's very bad. So, this and some of these processing steps can only go so far to removing some problems. Okay, and then I just mentioned group analysis. In the top row are, 5 or 6, let's say 6 brains from a given fairly homogeneous population, and they're all rigidly registered, so the ventricles are kind of in the same spot. Now, if you notice when you look up there, these brains have different shape, size, kind of structural patterns, things like that in detail. And so now, if I want to say, let me go to this location and have it be the motor cortex, I'd have to jump around in lots of different brains because all the brains are different shapes and sizes. This has nothing to do with intelligence or functional behavior. It's just an anatomical quirk, but it's also an annoyance for group analysis. So how do we get over this? What we'd like is to be able to say, I want to go to this location in a data set and have it be the motor cortex of every single person. So what we do is we take every person's brain and we treat it like Play-Doh, squish it, squash it, stretch it to a template brain, hoping that all the sulci and gyri of the same part of the brain line up well. So we define one particular brain as a special template, and we perform what's called nonlinear alignment that's really allowed to mush the brain around. And down here in the lower panel, these red edges you see are the edges of the anatomical template. And one for one, you can see where that brain has been squashed to here for each one of these. So you can see the amount of heterogeneity of what structure of the brain is where in the picture is greatly reduced. So we did that by, in some places, a larger brain is going to get squashed down, a smaller one will get stretched out, and we're hoping to make these sulcal and gyral patterns match, because that's where the gray matter lives that we care about. And the motor cortex lives, I'll embarrass myself, I'll say right here, I'm not entirely sure, maybe we'll go here, right? And so we're hoping that we align the motor cortex of every individual brain to sit in that same sulcus. That's the goal of this. And here's a picture of this nonlinear alignment happening in 3D. It's doing kind of a global fit and then mushing around and trying to get all these sulci and gyri to fit where they should be. Okay? There's fun mathematics behind it, but that's the goal of it. Any questions on this? Right. Okay, so this first part here, I just wanted to explain a little bit about fMRI, pluses, minuses, where it comes from, some of the steps that we need to insert into processing. So particularly motion correction and alignment are done on so many different studies. There's a lot of other subtle points that come along there. But the more you understand about the biology underpinning it, the physics of how that gets translated into your data, the realistic things and the annoyances and what to look out for, it helps you kind of with your processing steps and what to do to make sure your data is usable. Sometimes the distortions are at a level where things are fine and you can use your data. You maybe just avoid analyzing some parts. Other times it's totally crushed your data and you can't use this, or there's an artifact or something like that. So it helps to know what really are these good, bad, and ugly things. All right, and now what we transition to is what my group works on is the processing method. So we don't really acquire our own data. That's too difficult. We stay on the software analysis and design side to try to implement some of these things. So the alignment algorithms, motion correction, statistics, things like that. And in particular, trying to put this whole, I have raw fMRI data, I have beautiful processed fMRI data, connecting those two things in a pipeline. We have a tool for that called AFNI proc. So there are a lot of different software out there. There are different pipelines and choices. Everyone kind of builds ones to their own needs and desires. And so I'll just talk a little bit about this now. Any questions on any of this? It's quite a lot of stuff in a very short time. Are your brains now distorted with these geometric features? All right. One thing I'll just emphasize is something that like our group really tries to focus on. So we all like mathematics. It's great, it's powerful. There is nothing as good that you can do for your data and data analysis as looking at it at the beginning, the middle, and the end, and making sure that things are on track. So it really takes a lot of time. Many of you have just come here this summer. Here's a project, go, make sure the data is fine. And this may be your first time working with fMRI data, so learning all these things about what's good, what's bad, what's expected, what's out of bounds, that takes a lot. But hopefully you can ask people while you're here or whatever data analysis you're working on. This is just my soapbox for visualization in science. And we'll move on. Okay. So the things that we just looked at, this one, yeah. All right. So what I just mentioned was there's a lot of steps that we have to do, different kinds of alignment, EPI to the anatomical template, motion correction, maybe the anatomical volume to a reference template, all these things. And so putting together a pipeline is basically like thinking about a flow chart where you have major steps you want to do, and then maybe you have different options that you want to control with all of it. So this lovely thing here that everyone I'm sure loves to see is one giant processing command. These characters on the side here are called continuation of line characters. And so this is, you're providing a subject ID and some data sets. And this is the kind of thing that we like and we think is really pretty because it makes our life simple. I'll try to convince you of that now. It probably doesn't seem that way. Maybe a better way to think about it is this is one command, but it has little different pieces in it. So basically, the green part is you provide your EPI data, your fMRI data, and your anatomical data, and you say, this is my subject label ID. Then your blocks are what steps you want to happen. Do you want to do motion correction? Most people will. Do you want to align your data to a template? Maybe, maybe not, and other things like that. So you basically specify keywords for how you do it. And then for each one of these keywords, you can provide 0, 1, or more options to refine that behavior. And so that's why it's a long command, but actually the pieces aren't so bad. And the more you know about processing, the more you will be willing to make choices, or you can take kind of default examples and tweak those. But hopefully, if nothing else, this is a much more colorful version of the last one. So if I haven't convinced you that it's not too terrible to look at, then there you go, at least it's colorful. So I shouldn't even ask, but has anyone run AFNI proc in your summer while you're here? No, okay. Let the record show we don't know. Okay, so this command actually doesn't do the processing of your data. What this does is it actually sets up a whole processing script for you. And this is another, I'll take kind of a brief soapbox moment. When you're processing your data and when you're doing your analysis, you do want to know what's happening under the hood. And I know that things are new, there will always be new terms and things like that. But if someone says like, oh, what happened with your data? What was this choice about your blur size or whatever? It is nice to be able to know that, or to be able to go find it or work with someone to figure out what it was. And so that's why that first command doesn't just process your data magically and hand it back to you. It wants you to be able to know what happened underneath the hood. And so it actually generates a fully commented script. So I don't know how many people do scripting here? It's part of your summer or anything else, or programming at all. How good are you at commenting and writing really detailed but short and useful, pithy comments? Yeah, okay, okay. Well, I'm not great at it myself. This command does it automatically. So this is an amazing thing. This was written by Rick Reynolds in our group. He's a very good programmer, and he made this tool that will kind of script and program like him, which is great for the rest of us, because I wouldn't do this myself. So it's nice that you can not only see the commands that were run, but it will tell you what it's doing. So this is, your generation might not know that these things were harder before chatbots and all of this, but this used to be a harder thing. So anyways, all right. And then when it processes, it doesn't just give you your data, it also gives you like a report about it, about different things in there called quality control report. And so that's what I'll jump to now rather than looking at more code. So this is a command. This is from, we teach these classes called boot camps. This is absurdly large now, 1 second. There. So this is like a processing command from there. And again, this is probably, looks kind of ugly, but the main thing is that you can really break down and put in there what you want for every single step. So there's lots of choices. This is a task design for audio-visual, and you can choose how much to censor based on motion. You can provide labels for different stats and things like that. Anyway, so we're going to look at the output of that. When you run this command, if I pipe this through word count, there's 194 files here. This processing is not just giving you one output as a result. We like to keep all the intermediate results and things like that. So there's a lot of data sets, intermediate ones that you can check, a lot of text files kind of putting out useful information all there. What we're going to do is we're going to jump to the QC report and open that up. All the fonts are so large to do that. Okay. And so again, as you're kind of doing things and processing, science is entirely computational now, and almost every scientific field I can think of, you go through lots of different steps to get from A to B. I will just emphasize again, you really want to know what happened there and check and make sure that different steps work. So this particular iteration of AFNI-Proc ran start to finish. It had alignment as part of its processing steps. It didn't crash during alignment. Did it do a good job? We don't know. So the way you do it is you kind of check it. And the goal of this QC report is not just to process your data, but to help you be able to check things very quickly in there. And so these tabs up here kind of give you different things that you can look at along the way. So you might want to know that your original data was reasonable and not terrible. So we make these like systematic images for it, so you can check them out and you don't have to load a data set. You just look here and say, oh, the brain was mostly in the field of view. If you care about the cerebellum, you're going to be very sad seeing this, but now you know. Okay, so I was talking about alignment. This is the kind of way that we look at something like EPI, anatomical alignment. When you have two data sets, it's a little hard. In that earlier image I showed of the hot and cold over gray, how do you see where the features match up? It's a little tricky. So we use this kind of outline of one data set and see where the borders match up with the underlay there. So hopefully you can see the red outlines-ish, mostly, yeah. And there will be parts that don't perfectly match up here. That's probably due to the kind of skull stripping of the anatomical, but what we really care about are all the sulcal and gyral features matching up here. And the gray matter here is actually the brightest part, and the white matter is darker gray, confusingly, but all the sulcal and gyral features look like they really match up well. Where don't they match up well? Down here in the cerebellum, because our fMRI data didn't have a cerebellum. That's fine, and we know that. Where else doesn't it match up well? It doesn't actually match up well here, because for different reasons, our EPI essentially has no high signal here. Again, these magnetic field distortions. So here the cerebellum's out of the field of view, and now we also get this sense of like after alignment that there's just missing fMRI data there. Now we know. Okay? There are several features like this that you can really only check visually. There's a lot of computing power and things. I'm sure LLMs and image processing are going to be brought into this in the near future, but there really are things where you have to look at a picture to judge alignment, because you used your best algorithm to judge alignment and to drive it. Now you have to use your eye algorithm to judge it. Similarly, we had anatomical to template alignment. This looks good. Ultimately, what we really care about are the statistical modeling. And so I'm not talking about that really here, but this idea of what we started with 40 some minutes ago was I give you a stimulus, you can tell me what the bold response is, and you can pick it out. This time series, this one, this one, this one. This is letting mathematics do the work for us. This is what's called the full F statistic. So this is basically showing where our regressors of interest, those that are associated with our stimulus, where we predicted what our response should look like, where are those really good fits for our data? And the yellow is the best. So kind of here, 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 here. And this was an audio-visual task. I'm not a brain person, but someone told me these are mostly audio-visual parts of the brain. So at least I'm kind of happy. I don't think the subject fell asleep during the scanner and things like this. So this is a way that I can see, do things make sense from some kind of expectation I have about my data? I don't know much in the deepest sense, but I know an audio-visual task should give me some of these. And then the more research questions in here start getting into just the visual or just the audio. These are statistical responses for measuring where there's strong visual response. This is a more complicated task because visual is not pure visual. There's actually audio mixed in, which is why we see things here, but that's too much detail. I'll skip over it. But anyways, the idea is you can check your data systematically in this way. So whenever you're processing, you want to know, you want to set up your analysis how you want. You want to check the processing that was done. There's that script. You want to check the results and intermediate stuff. That's what this HTML try to have in it.
It's been a while since I've used statistically, but my understanding is that it's, this is for every singular participant. But
That's correct.
you run AFNI crock-pot like 20 participants at once? Like,
Yes, so the question, maybe the owl can hear this, but the question is, can you run this over a large number of subjects rather than just one subject? Yes, what we do is we put it in the script where the input variable is the subject ID that determines path. So it's all the same commands being run on everyone. Yep.
Yeah, then we just want QC.
Yeah, so actually I'll do a quick example of, so this QC, this is nice, we're gonna take 5-10 minutes going through it. In the boot camp, we spend maybe half an hour really getting into the details. Actually, I think a lot more. But how do you do this when you have 100 subjects? Do you have that much time in your life? You're young, you do, so that's fine. I'm old, I don't have that much time. How do I look at the QC in so many subjects? Actually, there are some features in these QC HTMLs where you can just blitz through everything very quickly and see. I'll mention that briefly. Yep.
Why are there colors outside the brain?
Good question. Why are there colors outside the brain? Because the error around this subject was thinking. Well, we gather data everywhere in a whole field of view, right? We put someone in a scanner and then we make this rectangular field of view and we gather data everywhere. There's a lot of discussions around this. We want to measure and do the modeling and see the results everywhere, even in the absurdity of the air around the brain, because we also want to know, are there other magnetic field things happening? So there's something called ghosting, where kind of low energy or low magnitude images, copies of your brain get kind of shifted, and so they also appear in your field of view, and you want to make sure that some of your results are not due to these. You want to make sure that alignment was really good. There might be other artifacts that you can discover that way. So we make the choice of presenting results in a what I call uglier way, seeing all this noisy stuff outside the brain. Another benefit is, as I mentioned, that fMRI data is noisy. You want to make sure that your signals and the things that you're seeing with your responses are kind of above the noise floor. So this inherently lets you see where that noise floor is. And this is just a single subject data. When we get to the group level set of things, we still model, we can still compare things there, but what we really want to see is that those things go away by the time we add more and more subjective data. We want to see that background magnitude decreasing and decreasing. If it's not, we've got a problem. And I've got a spam caller calling me. We want to be aware of these things. So we have kind of an engineering mindset. We'd rather know, is there any possibility there's a problem here? Let's check more stuff, even if it's a little aesthetically less pleasing. This gets into, if you notice, there are some very faded parts and some very bold parts. We don't like to depend on thresholds to say this is important, that's not. We want to see the modeling everywhere. And so we try to balance that as well. So there's a lot of layers here. I'll get really worked up talking about this topic anymore, but good question. Any other questions? Good. These are static images. Sometimes you want to know like what happened between slice 5S and 27S. There's a button here to run what's called knee view. So now you can click around and actively investigate anywhere in any slice that you want. This is telling you your coordinate location and your values and your underlay and overlay data. There's even this really fun feature here that I can hit the C key and clip it, and now I can move back and forth and see it in kind of a 3D picture and lots of different planes. It's very fun. I can also run AV, and I can see AFNI at work. So now I can load up the same thing in the GUI. Maybe it's getting there. There we go. And now I can browse it in the same way. So again, there are these kind of layers to how much you want to spend on your data, how much time you want to spend on your data. If you're happy with the image, great. If you're questioning it, you can really dip into it in a more detailed way very quickly without having to choose all sorts of values. Josh, I believe in his talk earlier this week, mentioned something called Instacore. And this is something where even for task data, but especially for resting state data, which I haven't really talked about, is something that is very useful. So the data that we're looking at here, the fMRI data, it has 3 spatial dimensions and 1 of time, right? And it's really hard to look at 4 dimensions of data all at once and to put all that information together. When we look at things like here, I want to see, you know, this is maybe the 3 dimensions of my data, different slices across the brain. Is it distorted? Is it the whole field of view? Fine. When I get into my statistical modeling, this is something telling me about time series by time series, how did my regression model look? That's all very useful, but nothing really ties together the space and time aspect. Instacore, and I'm going to click the IC button, is that feature that does that, and it's probably something I would call like the most useful QCing thing you can do. I won't trust data unless I can run Instacore on it. And so now what's been loaded up here, there's a description here if you ever run this. What this is I'm taking my full 4-dimensional time series, so the whole volume and all those time series are there, and now I'm able to hold down Control-Shift and click somewhere and see the correlation pattern from a little ball around where I clicked. And it's instantly updating correlation pattern, that's the Instacore. Now, again, this is task data, so at the moment I'll pretend like it's maybe resting state data. This is something I would want to check. If I put a seed in the motor cortex of one side, I generally want to see the motor cortex and the other side as a sanity check. There are tasks that separate this out, but anyways, this is often, you know, in general cases, that's something useful. If I click into white matter here, I don't, oops, let me not go there for one reason I'll get back to. If I click in white matter here, I don't see a big spread out correlation pattern. And again, you see little faint stuff all the way around because I've chosen to reduce the emphasis of threshold. I want to see anything regardless of what threshold I choose to emphasize. So I allow fainter stuff that's below my threshold to shine through. But I don't want to see white matter connecting everywhere. That would make me a little worried. So again, when you're analyzing your data, you want to make sure the good stuff is there that you expect, and you want to make sure that weird things that you don't expect really aren't there. Does that make sense? Okay, now let me jump back to where I happened to be before. Oops. And run Instacore. Where was I? Here. Now, this was a funny spot. I'm in white matter and I'm looking at correlation around. Just in general, whether you're used to fMRI or not, you don't really expect white matter time series to correlate a lot because those are not the functionally active ones, as is your 0th, 1st, and 2nd order effect. What do you see as you look across the 3 slices of my brain, though, in this part of the white matter? Actually, there is some kind of extended thing here. If you look in this coronal slice or this sagittal one, and if I click somewhere in here and move around just a little bit, there's kind of a finicky spot here that is going to show these high spatially extended patterns. This is the reason why I won't trust data. There we go. I won't trust data if I can't run Instacore. This is a weird spatial temporal pattern in the data, right? If I only look at the spatial part, I don't see this. If I only look time series by time series, I'm not going to notice it. When I look at these things together, I see this. There's no cortex of the brain that looks like this, I guarantee you. This kind of candy striping of correlated, anticorrelated, correlated, anticorrelated is some weird artifact. To date, we still don't know exactly what it is, but it's in this bootcamp data set. We never noticed it until we did a QC project where we found actually something similar, and so we added this feature in to be able to click on a button and really dip into the data like this. And now we've seen this in a surprisingly large number of data sets. I will note that this is not from NIH, so it's fine. All your data is fine. But we've seen this in huge fractions of public data. It's really worrisome. And so, again, acquiring data is super hard. Dealing with magnetic fields, super, super hard. And it's just this point of whenever you're processing, there's a lot of subtlety to stuff. So, again, just kind of zooming out a little bit, whether you're doing fMRI or whatever tool you're using, you really want to know what you're doing, what's expected, what's unexpected, look for unexpected weird things to make sure they're not there, and have this kind of holistic view, because otherwise they're just weird things lurking in your data and you won't know about them. So in this way, you kind of want to always connect what you're doing with these things. And as you go along your science career, you want to develop ways to make this as streamlined as possible. So that's been one of our goals, is not just processing the data, but to try to connect these things to let people kind of dip into them, because it's so necessary, because it's just a hard set of data to work with. Any questions?
So just to sort of get to recap what we're looking at there. We're in, looks like we're fMRI data. So if you're in the white matter.
Yep, we're in white matter.
And you're just seeing this weird kind of like pattern, like vertically or what?
Yeah, so specifically what we're seeing is a very narrow column.
Yeah.
And what I'll do is I'll jump to a different way of looking at this. So I'm going to jump, this is process data that's been warped to standard space. I'm just going to run Instacore in raw data here. Let's see if I can. And the funny thing about this artifact is it is a very narrow column. Well, okay, let me cheat further, because this is something we actually automatically warn about this now. So we found this as such a big problem that basically what's happening is you get these very weird narrow columns through your data. So the data is acquired slice-wise, these axial slices, and for whatever reason, at one point in your XY grid in those slices, you have something very bad happening. Again, still trying to narrow it down, but let me just jump to this location. Instacore set, there. So we can identify these where these columns happen in the raw data. This is not a correlation pattern you should ever see. So this is, again, this is tacit, but even in REST data we see it, and different things like that. So yes, it's a weird feature. The data is acquired, interleaved. That's kind of why the pattern is a bit interleaved, I think. But yes, so it is weird.
What it looks like the case base.
Yep, it's true. So thinking about it, slice-wise, if you, I think this is like so if you have a spike in image space, in case space, from a magnitude point of view, if you just have a single spike and you do a 2D FT, it's like adding to the magnitude everywhere.
Right.
So somehow I expect it's related on a slice-wise level to something like that, but I don't know.
No, kind of curious what you guys figure out.
I will be too. Yep, happy to hear ideas. This is why it's called science and not history. Although I like history. Any other questions? Do you want to do more fMRI analysis now or less fMRI analysis after this talk? Are you scared off of it or encouraged? No. All right. All right. I will stop the recording.